All right, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 167th seminar, and I call it a University of Warwick Day. We are facing many global problems, including wars, economic instability, human rights, food inequality and shortage, energy issues, and also sustainable production, pollution, and the climate crisis. Among these, the climate crisis due to pollution that include, plastic and other pollution, has shown that we are approaching, unfortunately, the tipping point. For example, we saw more than 2 inches of rainfall in an hour in New York City, a huge number of deaths, unfortunately, in the devastating Central Texas floods earlier this month, and also another tragedy due to the European heat wave this month. Such extreme weather events are only going to intensify if no immediate actions are taken. Fortunately, many engineering biology researchers have been working to solve the pollution and climate crisis issues, and I hope we will solve these global problems together. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Tim Bugg. He is a professor of chemistry at the University of Warwick, the United Kingdom. I have known him for a while because I have worked on lignin valorization since 2012, but I only recently met him for the first time when I visited Warwick in, I think, 2022 to give an invited talk. He is a true pioneer of lignin enzymatic degradation and renewable chemical production. Fortunately, our global center circle has him as an investigator, and as a center director, I am so happy to collaborate with him internationally to solve waste issue that causing a lot of problem, including, of course, the climate issue. Tim, thanks so much for your pioneering work on lignin enzymatic degradation, and the virtual podium is all yours now, and thank you again.
Right. Thank you for your kind introduction. So I'm just going to share a little presentation. Uh, right. Can you see that?
Yeah, looks good.
Yep.
Go ahead. Thank
Okay.
you.
So I'm just going to give a short introduction to the lignin degradation research that we do. Um, and so first of all, so this is about enzymatic and microbial conversion of lignin to renewable aromatic chemicals. And so, first of all, just say something about Warwick. So, in the top left, that's the University of Warwick, just on the edge of Coventry in the Midlands in England. Fairly green campus with some nice buildings around campus. And there's lots of history around Warwick. So, near us is Warwick Castle on the bottom left. And I live not far from Stratford upon Avon, a historic town where William Shakespeare was born and where the Shakespeare Theatre is. There's lots of history around. So, if So going on from the introduction from Taysok is if society is going to move to some kind of low carbon economy, we've got to reduce CO2 emissions. I think that's obvious really to all the scientists around the world. And that means that we have to move away from our dependence on crude oil for making both fuels and chemicals. And there are different strategies for doing that. The strategy that I'm working on is to try and utilize plant biomass to convert that into fuels and chemicals. So the idea is that you can take a crop, you can take the food part of that crop, and then use the non-food lignocellulose. You do a thermochemical pretreatment on that, and that generates three fractions: lignin, cellulose, and hemicellulose, which are the three components of lignocellulose, and then convert those into fuels and chemicals. So in the top right, you'll see the chemical structures of these components are quite complex. So unlike an oil refinery where you're starting with very simple ingredients, you have to deconstruct these and essentially remove functional groups to convert them into useful chemicals. So briefly, from cellulose, that can be converted via glucose into bioethanol, which we can put in our cars. So that provides transport fuel. But glucose and xylose can also be converted by fermentation into various chemicals, including organic acids like lactic acid and succinic acid. They can be converted into useful products like polylactic acid, for example. But lignin is the big unsolved problem in the biorefinery concept. But in principle, it could be converted into high value products that would add value to this whole biorefinery concept and be very useful chemicals in their own right. So a little bit more about lignin. It's an aromatic heteropolymer. It's heterogeneous and consists of aryl C3 units that are linked together with a variety of different linkages, the most common of which is the beta aryl ether. So you have an ether linkage between the two units. And then other kinds of linkages sometimes have carbon-carbon bonds. So these linkages are not susceptible to hydrolytic cleavage. And that means that lignin is really tough material to break down. It's also it tends to be insoluble, somewhat inert. So it's a really tough nut to crack. There are some variations in the substitution pattern between softwoods, which contain mostly G units, hardwoods, which contain S and G units, and then grasses, which contain some of each of these three different types. So it's challenging material to break down, but it is a potential source of aromatic products. It's the most abundant source of renewable aromatic carbon in the biosphere. So briefly, the two strategies that we study in my group are either trying to find enzymes that can break down lignin, and my group specifically, we look at bacterial enzymes that can break down lignin, and then try and apply those in vitro to make high-value products. Or you find a lignin degrading microbe and then you do metabolic engineering on it. You engineer that microbe so that it can break down the lignin, but you interrupt or you redirect the pathways in such a way that it makes a useful product. So two quite different strategies. They each have their pluses and minuses. They each have some practical problems. So I'm just briefly going to give you one example of each. So this is the first bacterial lignin degrading enzyme that was discovered that we found in my group in 2011. Peroxidase DIP-B from Rhodococcus jostiae, RHA1, which we found essentially via bioinformatic approach from the genome and then confirmed by gene knockouts. We found that the enzyme was active against different types of lignin, polymeric lignin, and then we used lignin model compounds to determine which bond was actually being broken. It's the alpha beta bond in this dimer. And then we use stop flow kinetics to pose the question, does this enzyme preferentially react with the lignin dimer, or does it react with manganese? We knew that it could oxidize manganese, and actually it can do both. So you can observe the reactive intermediates, the iron oxo compound 1, and measure rate constants for its reaction. So it's a kind of bifunctional enzyme. And together with Lindsay Eltis's group at UBC, we determined the crystal structure of this enzyme. You can see the heme cofactor, and my group and Lindsay's group have both done some protein engineering around the heme binding site of this enzyme. So that's just one example. We found a number of other enzymes. And then the other strategy is the metabolic engineering strategy. I'll just talk about one of these stories. So this is a collaboration with a company called Biome Bioplastics in the UK, and they are interested in replacements for terephthalic acid, which is used to make PET, which is found in drinks bottles. Lots and lots of things are made of PET. They are actually more interested in PBAT, which is biodegradable, whereas PET is not normally biodegradable. And the other components of PBAT you can make via fermentation from natural sources, but not terephthalic acid. So we were trying to make aromatic dicarboxylic acids, and we knew that lignin degradation proceeded through this intermediate protocatechoic acid, which normally is cleaved between the 3 and 4 positions via protocatechoate 3,4 dioxygenase and goes down this beta-ketoadipate pathway. But I knew that there were other enzymes that cleaved the ring in different places. So either between the 2 and 3 position or the 4 and 5 position to make extradiol ring fission products. And it was known in the literature that you could cyclose those with ammonia or with ammonium chloride. And so in principle, you could zip up the ring again to make a pyridine dicarboxylic acid. And this seemed a little bit far-fetched, but it worked remarkably. So we inserted these genes into Rhodococcus jossii, RHA1, using a expression vector, and we observed the production of these metabolites. And optimizing that, this was in 2015, we were able to get between 90 and 125 mg per liter after about 7 days. So it takes time to break down lignin, and that is one of the challenges. So that was an interesting breakthrough. And since then, we've been trying to optimize the titer and the productivity by deleting the competing pathway and by inserting the genes onto the chromosome and overexpressing lignin oxidation genes. And in 2021, we were up to about 330 mg per liter. And since then, we're up to about 500 mg per liter, which is progress, but we need to get grams per liter to be commercially viable. So we've still got some way to go, but we're definitely making progress. And so just to summarize, what I see is the challenges and opportunities in the lignin field. Lots of challenges. Lignin is hard to degrade. There's lots of different types of lignin, and they all behave differently. The enzymatic lignin degradation, although fascinating, because I'm an enzymologist, there are practical problems that you can get repolymerization and you get high molecular weight material, which I haven't got time to go into. And you also get mixtures of products. And then the metabolic engineering, there are challenges there that we don't fully understand all the lignin degradation pathways. And as I've described, it's quite hard to achieve the high titers that you need for industrial take-up. But lots of opportunities. So there's definitely a societal need for low carbon routes if we're ever going to get to net zero. There's lots of lignin available from various industries around the world, pulp paper manufacture and bio, uh, biofuel production, biorefineries around the world. There's lots of aromatic chemicals that are used in the chemical industry, and those producers are now starting to think about where are they going to get their chemicals from in the future, and potential to kind of link this up through primary metabolism to make other things like natural products, for example. So lots of opportunities. And then Tae-sok said, I should give a little advice to young scientists, which is very interesting to think about. So, why did I choose to do research? And this goes back to when I decided to do a PhD in Cambridge many years ago. So, really interested in science, fascinated by science and the idea of discovering things for myself. Um, I was always quite a practical person, a problem solver, and I enjoy solving problems in the lab. And I've always had lots of ideas, which is something that you need in research. So I enjoy following up my own ideas, but don't want to give the idea that research is all easy. Research is quite hard, and every researcher will have a time when things don't work for them. So it's good to have some other interests. So, for example, I used to play volleyball and I used to go cycling. So this was me somewhere in the French Alps. And then when I stopped doing that, I got out my violin that I used to play at school, and I played an orchestra and also with a Baroque band. So, if your research isn't going well, go and do something else. And then the next morning it won't seem so bad. And you think, okay, go again, and then you'll solve it. And then why did I decide to be an academic? So I was, yeah, having done a PhD and a postdoc, I was quite fascinated by enzymes. I had lots of ideas, which is you really need as an academic. And I also enjoyed teaching chemistry. So in the UK, the vast majority of research is done at universities. And so most of us do research and teaching. Um, and I've always enjoyed doing that. And one of the things that led me to do was to write a book. Well, I did that when I was at Southampton. And one of my projects for this summer is the fourth edition of the book, which Wiley talked me into. Um, but again, I don't want to give the impression that this is all easy. It's quite a challenging job. So getting research funding is really challenging. Sometimes getting papers published can be really challenging. And you have to manage a research group, and there's lots of things that come with that. So managing people, personnel, finances, safety, lots of admin, etc. So it's a bit like running a small company, really. There's quite a lot of responsibilities, a lot of things to think about. And then finally, how to choose research projects. And I think when you start as an academic, this is a really important question. So academic novelty, really important. And so for me, I was fascinated by enzymes that catalyze unusual reactions. They've always fascinated me and they still do. But I've tried to choose areas that I think one day would benefit society in the future. So biotechnology, initially for bioremediation, but then more recently, biomass conversion to renewable chemicals. I also do some work in medicinal chemistry, so enzyme targets for drug discovery, powerful motivator that the work that you do might one day lead to the discovery of a drug that might save someone's life. Um, Something that I've found over the years that some industrial relevance is very useful. So we have a scheme called case studentships where an industrial partner puts 10% of the money into a PhD studentships. And if you're able to do that, then it's a bit easier to get a PhD student. So that's something I've found over the years. And then I like difficult problems. Lignin is certainly a difficult problem, but you also need to demonstrate that you can actually complete pieces of work and publish, and particularly at the start of your career. And my dad, who was a physicist, once told me that finishing things is a skill, and it is. Yeah, you need discipline to actually finish off pieces of work and demonstrate that you can deliver on projects. But yeah, Thank you for the opportunity.
Thank you. This is the fantastic talk and also the advice. I follow your work. Actually, your first paper you just described that is actually guiding paper, for my group for more than 10 years. And then these advices all touch my heart because I experience everything you may just mentioned. It's very difficult, very challenging, but the reward also huge after you enduring all years of failure, I guess. So this is a wonderful advice. Thank you so much. All right. Now the main speaker of today with a longer introduction. Dr. Erin Garza received her PhD degree from Northern Illinois University, where her research focused on engineering microbes for alternative biofuel production, such as biobutanol production. While she was a graduate student, she also worked at Monsanto in the entomology department studying western corn rootworm. Once she received her PhD, she began working at the J. Craig Venter Institute as a postdoc in the Environmental Sustainability and Synthetic Biology department. She continued to work as a staff scientist at the J. Craig Venter Institute, where her research now focuses on deep sea plastic degrading microbes and microbiota, as well as studying the effects of plastic pollution on the environment. I'm also fortunate to have her in our Global Center team working together on plastic degradation problems. That is obviously very important global problem we have. Also, I want to congratulate her because she very recently promoted to assistant professor from staff scientist. So now all team already mentioned to us, also applicable to her because she now starting very exciting but challenging journey in the future. So Erin, I'm so glad, finally promoted to assistant professor and congratulations again. And thanks so much for your time today. And please take it away. All yours now. Thank you.
Right. Thank you, Tae-suk, for that introduction. Let me just share my screen here. Okay, are you guys able to see that?
Yes.
Okay, great. So today I'm going to be discussing my research, which focuses on the microbiome of plastics, otherwise known as the plastosphere, in deep sea environments, and how to identify and test microbes with plastic degradation capabilities. So why look for plastic at the bottom of the ocean? There's plenty of plastic in easier to access places. Well, the deep sea is a naturally nutrient-limited environment, but most marine plastic pollution, which is carbon-based, eventually ends up on the ocean floor because it's either denser than water and it sinks, or it gets weighed down over time due to biofouling. Now, plastic has been in mainstream use since 1907 with the creation of Bakelite. So that means that plastic has been accumulating on the ocean floor for decades, and microbes have had over a century to evolve to break down some of these plastics as a food source. Now, the ocean also contains a lot of extremophiles that have had to evolve to survive things like extreme temperatures, high pressures, high UV radiation, or a complete lack of light, and even high salinity. So maybe plastic degradation is their next adaptation. And it wouldn't be unheard of to find plastic degrading microbes in the environment. It doesn't happen often, but it is starting to occur with more frequency. One of the most well-known examples is the microbe Edenella saccaiensis, which degrades PET. It was originally found around a recycling center. I think it was in one of the outdoor holding areas for all the plastics. So these heavily polluted areas are selecting for organisms that have the ability to utilize plastics as a nutrient source, or these organisms are evolving to be able to degrade some of these plastics. So we're kind of living in a very interesting time right now because we've developed all of these new polymer compounds, and now we're watching this evolution take place of these microbes being able to break them down. So how am I getting these samples? Accessing the deep sea is very difficult. Well, I was able to use the Alvin submersible and a deep autonomous profiler, otherwise known as a DAP, to collect water and sediment samples from the surface around 10 meters down to depths of 8,000 meters in the Puerto Rico Trench and the Mid-Cayman Spreading Center. Now, these samples were divided into two portions and analyzed to determine native microbiomes and dissolved organic carbon levels, or DOC levels, which is basically going to tell you how much carbohydrates, proteins, lipids, those kinds of things are present in the water. And so this was used to create a depth profile of the water column. Now, the other portion of the water was used in plastic incubation experiments. I was working with 4 different types of plastic. 2 were biodegradable, so I had cellulose and polyhydroxybutyrate, or PHB. And then I had 2 non-biodegradable plastics, high-density polyethylene, HDPE, and polyethylene terephthalate, PET. Now, these plastics were cut into about 25 milligram pieces and sterilized with ethanol. Then they were added to a glass bottle with 100 mils of the collected seawater, or a glass petri dish with the top 2 centimeters of a sediment core. And you can see pictures of those at the bottom of the slide here. So the bottles and petri dishes were covered with foil to ensure that any degradation that occurred was due to microbial degradation and not from photodegradation from light. And this also helped to protect these deep sea microbes from UV stress. Now, the surface microbes were then incubated at about 26 degrees Celsius, the deep sea samples at 4 degrees Celsius, and this went on for 5 weeks. And then metagenomics and metatranscriptomics were then performed on the plastic biofilms to determine what was selected for as a result of the plastic. And the water was then analyzed again to determine final DOC concentrations to look for indications of plastic degradation. Now, these are some examples of DOC results that we had from our samples. As a reminder, the first time point was taken when the water was initially collected and had not yet been exposed to the plastic. So this is kind of our time 0 time point. The second time point was taken after the water had been incubated with the plastics for 5 weeks. Now, during that entire incubation period, no additional nutrients were added and no water changes were performed on the incubations. So all that was in there was the plastics. That was the only addition that we made. So large increases in DOC would not be expected unless microbes are actively growing and releasing compounds, or if the plastic itself is releasing some type of carbon compound. Now, based on the graph seen here, we can see that regardless of depth, all of the plastic incubations showed an increase in DOC. Now, typically the PHB and cellulose samples showed a larger DOC increase, which is kind of to be expected since they are biodegradable. We would see microbes actively growing in these incubations. However, some of the non-biodegradable PET samples showed about a 100-fold increase as well. So this increase, it could be due to degradation, or it could be due to leachate from the plastic. So at this point, further testing was required to determine if degradation is actually occurring. So next, we imaged our plastics to look for the presence of biofilms and signs of degradation on the plastic. SEM analysis was performed by my collaborator, Erik Zettler. He's at the Royal Netherlands Institute for Sea Research. And so if we look at the first panel here on the left, that shows a piece of PHB at varying magnifications. So the image at the very top, that is at 100x, and we can kind of see a little bit of an outline here of a biofilm that's forming on the surface of that plastic. As we increase the magnification, we can start to see individual cells and kind of the biofilm protein matrix. And then all the way at the bottom, that's 10,000 times magnification. We see a variety of cell types, including these large kind of corn cob looking cells. I'm not sure what those are, but they look really cool. We also see an abundance of cells on the cellulose. That's up at the top left here. That was incubated in water from 5,600 meters deep. And we see complex biofilm formation on the PET sample from 3,000 meters. So we know there are cells present on all of these plastics. It's still kind of difficult to determine if they're actually degrading the plastic. We do see some pitting seen like on the cellulose sample. Like if you kind of look closely, it looks like some of these cells are kind of digging into the plastic. That's because they're actively degrading it and kind of making a little hole for themselves. But 5 weeks wasn't really a long enough period of time to see if this is occurring on PET and the HDPE samples. So next we performed FTIR, or Fourier transform infrared spectroscopy, to detect degradation on the plastic itself. So FTIR will provide you with a chemical profile of your sample. It's essentially measuring the abundance of different functional groups. So changes in abundance of key functional groups is an indication of degradation. So for example, if we look at the PHB spectra in the top panel, we can see that the PHB control, which is a virgin piece of plastic that's never been exposed to any enzymes or bacteria, that's represented by the blue line here. That does not have a peak at 3,400 wave numbers, but the 3 experimental samples do. So this indicates that there's an increase in the presence of OH groups in those samples. So if we look at the chemical structure of PHB, it is made up of repeating units of 3-hydroxybutyrate. So the only way we would detect OH groups is if the polymer chain was being broken down into the monomer form, the 3-hydroxybutyrate, creating that 3' hydroxyl group that's seen here. So based on the literature, as PET is degraded, it will also show an increase in OH groups, which we can see in one of the samples in the bottom panel. It does have a definitive OH peak. So this is kind of helping to tell us which samples are showing signs of degradation and which may contain these plastic degrading microorganisms. So we know which samples to kind of focus on and move forward with. We also have FTIR spectra for the HDPE and cellulose plastics as well, but they all share the same kind of general principles, so I'm not going to go over each of those. So now that we know some of our samples may contain these plastic degrading microbes, we will look at some of the bioinformatic analyses of the samples to determine what species are present. So this is a taxonomical profile heat map. It was created using SynBYSS, which estimates relative abundance of taxonomy using metagenomic profiling. Now, the reason we use SynBYSS instead of something like Kraken 2 is because it's faster and more accurate, because it's not biased towards more abundant taxa since it uses a reads-based approach. So if you look at the bars on the top of this heat map, you can see the samples are identified by incubation time. We did sample a few of these after 1 week, the majority at 5 weeks. And then you can also see they're classified by location, so Mid-Cayman Spreading Center, Puerto Rico Trench, and then also by sample type. So we have water column samples, which are all kind of underneath this blue bar here. And then the rest of these are all plastic biofilm samples. And I'm going to ask that you just kind of ignore this one lone water column sample here. The sequencing on that one didn't come out. We need to resequence it, so that's why it didn't cluster with the other water samples. So each of the red lines in this heat map corresponds to a different taxa that is present in the sample. So based on this, there is a clear distinction in the taxa that are present within the water column. So again, over here on this side, versus what's seen enriched for on these plastic biofilms, which is kind of the rest of this heat map. And there's a huge drop in diversity on the plastics versus what is seen in the water. So next we look for correlations in our metagenomic profile data based on depth and plastic type. So to do this, we graphed our data on PQA plots, which measures dissimilarity among data points. Now, the plot on the left is analyzing samples by depth, and the plot on the right is analyzing by plastic type. If we look at the depth plot, we can see some distinct clustering of samples of the same depth. We kind of have a 4,200 meter cluster here, a large 3,000 meter cluster here. There's a bunch of 10 meters clustered there. But if we look at the plot on the right, we don't really see any patterns between the plastic types. Everything's just kind of spread out. So this verifies that sample similarity is depth dependent and not really affected by plastic type. So what we're seeing is kind of a settling effect where samples will have more similar metagenomic profiles if they are from the same depth. So this is likely due to environmental conditions like pressure. Not all microbes can withstand high pressures. So as you travel down that water column, the diversity of the microbes is going to change. So anything sampled from a particular depth will have more similarity to other samples from that same depth. Now, this is a chronoplot, which shows what taxa are in a sample and at what abundance. This is a time zero sample of the nepheloid layer. Now, the nepheloid layer is the layer of water just above the ocean floor in the deep sea. In this case, it's around 5,600 meters. It typically has a higher concentration of sediment or suspended particles, and it can also have higher nutrient levels. Now, this plot is on the order level for readability, but because it is still kind of hard to read, I'll just kind of sum up what's in the plot itself. 17% of the organisms present in this sample are archaea, with a high abundance of nitrososphoralis, which are ammonium oxidizers. And the rest are prokaryotes, with a major phylum consisting of proteobacteria at 39%, actinobacteriota at 15%, and unassigned bacterial taxa at 13%. So this is telling us that there is quite a bit of diversity in this water column sample, and this plot is kind of a graphical representation of the organisms that were in the metagenomic profile heat map that we just saw a minute ago. Now, this plot is representative of what we saw on the biodegradable plastics, so the cellulose and PHB plastics in water from the same location. So what we're seeing is that there are no longer any archaea present, and there is an enrichment of proteobacteria. It's now jumped from 39% of our sample to 88% of our sample. Now, the majority of the organisms that are here are Rhodobacter, Alteromonas, and Pseudomonas. And this does kind of make sense since all of these organisms like to attach to particles, and we commonly see them growing in biofilms. Now, this plot represents the non-biodegradable plastics. Again, this is taken from the exact same location, nepheloid layer in the Puerto Rico Trench. There's still a lack of archaea on this sample, but the microbiome is now made up of 73% proteobacteria and 14% actinobacteriota, which we didn't see on the biodegradable plastics. Now, one thing to note is that many of these samples contain Myxococcus, which do predate on other bacterial species. And there's also a variety of bacteriophages on all of the samples that we had. So kind of the key thing to take away from these analyses is that these plastics are enriching for specific bacterial species and causing a decrease in overall biodiversity. And they're also kind of creating these unique mini ecosystems that are significantly different than the surrounding environment, which may have the potential to affect local ecology. Our bioinformatics data was further analyzed using the VIBA pipeline, which was created by a former colleague of mine, Dr. Josh Espinoza at JCBI. VIBA is an automated modular bioinformatics software suite that is assembly-centric, and it can be used for prokaryotes, viruses, eukaryotes, and archaea. Now, this pipeline was essential to analyzing our data because it gave us a genome-resolved blueprint of what organisms and genes are involved in plastic colonization and also possibly degradation. Now, VIBA was used to analyze our transcriptomic data to determine what genes were actively being expressed in the plastic biofilms. Now, the corresponding proteins were then blasted to try to determine their function. The issue with using BLAST, however, is that the results don't always give us a definitive identification. So in the examples that I have listed in the table on the right, we can see that BLAST determined that we had two different hydrolases. One was from alkanovorax and the other from Halopseudomonas. And now, based on the BLAST results alone, we wouldn't really know what these hydrolases are doing. Are they actually involved in plastic degradation or not? We don't know what the substrates are for these proteins. What are they acting on? All we know is that it's an alpha beta fold hydrolase or a dienolactone hydrolase. It's not very informative. So when you have vague identifications like this for thousands of different proteins, screening for potential gene targets can become a very daunting task. So kind of where do we go from here? Well, we ended up turning to the Plastic Degradation Database, or Plastic DB. This is a database containing experimentally verified plastic degradation genes. So genes that are involved in plastic degradation tend to be classified as things like cutenases, lipases, alkane hydroxylases like monooxygenases, and lactases, which are copper-containing oxidizing enzymes. Now, the one issue with using this database is that it only contains known plastic degrading enzymes, and there aren't really that many. The last time I checked the database, it only had about 219 known enzymes in it. So it can still be difficult to identify and compare novel enzymes using this method, but it still seemed promising for identifying proteins of interest. So we ran our protein sequences against the plastic degradation database to see if it could provide us with some more definitive gene targets. So when we did this, we had over 2,600 positive hits to the database. We then filtered the results to determine the best hits by looking for proteins that had a bit score of over 50. They needed to be at least 100 amino acids long and have a percent identity of at least 45% to a hit in the plastic degradation database, because that would just kind of ensure that the protein we had is more likely to have a similar activity to the protein in the database. So originally we had the two proteins classified as hydrolases by BLAST, but when we compared those to plastic DB, we see that the first hydrolase is actually about 70% identical to a hydrolase from Alkanovorax borcumensis that has been shown to be involved in PLA degradation. And now our second hydrolase protein that we were looking at is actually about 75% identical to a cutinase protein from Pseudomonas oleovorans, which breaks down PET. So this is a lot more definitive. I would feel much more comfortable cloning these two genes now and testing for plastic degradation activity, because based on plastic DB, this is saying these have very high similarity to known plastic degrading enzymes. So this is kind of how I was trying to figure out what are going to be the best potential targets to work with. So this really kind of created that jumping off point for me for screening these proteins. So now that we have a list of hundreds of potential plastic degrading enzymes, each of them will require validation. So in order to do that, we're going to use two different approaches. The first is a high throughput approach, where we're going to have each gene individually synthesized using a grant that we have through JGI. So each gene will be inserted into its own PET vector. It'll have an N-terminal tag so that we can purify that protein back out. And then we will incubate it with its target plastic and then test the plastic for degradation. And the plasmid map that you see at the bottom of the slide there, that is an example of one of our PET vectors that's being designed. The highlighted portion is one of our plastic degradation genes that we're testing. Now we're also using a individual gene expression approach, where we're going to clone the most likely or proteins that have the highest potential to show degradation into the organism Alteromonas macleodii, and then test for degradation ability. So this is going to give us the opportunity to validate our high throughput results, make sure that we're seeing the same things. And it'll also allow us to evaluate the native secretion peptides that a lot of these proteins have. So we're not able to do that with the high throughput method because the N-terminal tags are on there. So those native secretion peptides wouldn't be utilized. So this will allow us to do that. For the individual gene expression approach, we are utilizing the loop assembly method. For those of you not familiar with loop assembly, it is a species agnostic system that allows for quick, easy assembly of transcriptional units and pathways. I designed and incorporated an Alteromonas cloning backbone into the system and then created a library of Alteromonas parts that could be utilized to generate our transcription units. So luckily, we ended up finding out that Alteromonas is actually very similar to E. coli and is able to utilize a lot of E. coli genetic elements like promoters, ribosomal binding sites, terminators, and we're even able to codon optimize all of our genes for E. coli, and Alteromonas doesn't seem to have a problem expressing those. So that's been really nice. I also redesigned the blue-white screening system in the plasmid backbone by replacing the LAC-Z gene with a blue chromoprotein. So colonies will grow up blue without the addition of additives like IPTG and XGAL. And so the picture in the lower right-hand corner here, this is an example of E. coli expressing a purple chromoprotein. So you can see that they do kind of produce this very bright, vibrant color. We chose to use the microbe Alteromonas macleodii to host our plastic degrading proteins because it's also a marine microbe. So we wanted to use a host that was going to be from a similar environment to where all of these proteins were sourced from. So Alteromonas is also ubiquitous in marine environments, except for in polar regions, but there are even deep ecotypes. And it's also a strong biofilm former. So this is especially useful if you're testing if the cells are breaking down a solid substrate, because those cells are going to be in direct contact with the plastic, and so they're secreting their enzymes directly onto the target. Versus, let's say, if you had a planktonic cell and it's just kind of floating by your plastic, kind of spitting enzyme out, it's not really going to be an efficient way to test if those enzymes are actually working on the plastic. So on the right-hand side here, we could see a couple of confocal images. This is of Alteromonas. That's what all of these little green dots are. Those are Alteromonas cells. You can see they form this nice, healthy, thick biofilm layer on this piece of plastic, which is seen in red. Now, because we're going to be performing a lot of genetic engineering in Alteromonas, we needed to make it into a model organism. And so we did this by developing efficient cloning methods. So we use conjugation typically for Alteromonas. And we created a library, as I had mentioned, of genetic parts that are compatible with this loop assembly cloning system. And so this is currently where we're at with this project. We're cloning all of our gene targets to validate their predicted activity with the goal of kind of identifying these new highly efficient plastic degrading enzymes so that we may be able to improve those enzymes further or utilize them for recycling. So to kind of sum up, this project has shown how to translate an ecology study into a synthetic biology study with real-world applications. By utilizing bioinformatics tools and pipelines like Biba to analyze genomic and transcriptomic data, we can gain knowledge on what species are present in a sample, their operon design, expression profiling, and their community structure, which is especially important for things like plastic degradation, since it's often a community that degrades a plastic, not a single species. And in this case, I now have a blueprint for designing a synthetic community of microbes that can now be used in recycling facilities to help improve our recycling capabilities, thereby preventing more plastic from entering the ocean in the future. So to continue this research, I would like to perform some more deep sea plastic microbiome sampling. I want to kind of target more unique marine environments across the globe. So looking at things like petroleum seeps, polar regions, hyperpolluted locations, petroleum seeps specifically because plastics are petroleum based. So these microbes that are living in these regions are constantly kind of in contact with very similar compounds. So they may be more likely to be able to degrade these plastics. Hyperpolluted locations, because of what I had mentioned at the beginning of this talk, Just that these areas, because there's so much plastic there, they're kind of selecting for these microbes that are able to break them down. So we may have a greater chance at isolating some new plastic degraders in these areas. I also want to collect my deployed C cubes and deploy more C cubes. So a C cube is what you see in the photo on the right-hand side here. These I am deploying in collaboration with Dr. Ashley Burkett at the University of Oklahoma. This is a project that she had started. She was originally designing and deploying these to attract foraminifera. That's what she studies. But then we kind of teamed up and we were using these to also study plastic microbiomes. So we attach a bunch of different types of plastics to the outsides of these cubes, and then we deploy them to the ocean floor. So this is an example of one of our cubes that is now sitting in the Puerto Rico Trench. And so these are long-term in situ experiments. What's really nice about these is that we can we have an exact idea of what's going on with the experiment. We know the complete incubation time when it was deployed, when it was collected, and we also know exactly what plastic types are on the cube. So when you go out to a location and you're trying to just find plastics and sample what's already there, um, you have no idea how long that plastic has actually been there. It may be hard to get an identification of that plastic just based on the amount of degradation that it's gone through. It's been exposed to UV degradation and everything. So this is a lot nicer. This is very controlled. We know all of the variables. And because it's in situ, this is going to help us select for piezophiles. So organisms that prefer to grow in higher pressures. So this is something that's probably missing from my current metagenomic profiles because we weren't able to incubate our samples at in situ pressures. That is very, very difficult to do. So if we're able to go back and collect all of our in situ experiments, we should be able to now sequence and see all of these other organisms that were probably lost from the current sample set. So with that, I would like to thank all of my team members and all of my collaborators and of course my funders. And I'll take any questions.
Thank you. Fantastic. I want to ask Tim whether he has question or comment.
Yeah. So one sort of general question, the the microbial strains, the microbes that are present, will many of those be culturable in the lab, or are they generally not culturable?
Um, so the ones that we're seeing, like the alteromonids, the pseudomonids, a lot of those are culturable. Those don't seem to be obligate piezophiles. Those are just kind of surviving in these areas, or maybe they are completely fine with growing at those depths. But those can be cultured. If it was a piezophile, then probably not. Maybe they were still kind of lingering in our samples, but I doubt that if we were to try and grow them up, that they would be very happy with us growing them at atmospheric pressure. But I think it is possible because our incubation bottles that we had after that 5 week period, some of them were very cloudy. So when we initially put the water in the bottles, they're very clear. You don't really see any growth. But then after that 5 week period, you see a lot of visible biofilm. So they're growing that I would say that those were all culturable.
Yes, because we've done some screening for lignin degraders from soil and also from waste. But there we're generally looking for particular strains and
Mm,
then we sequence the genome. And so, yeah, the advantage of your metagenomic approach is that you sequence everything, whether it's culturable or not. Yeah.
yeah, yeah.
Okay. And then, yes, I mean, you mentioned at one point, how do you find unknowns, which is then
Mm.
much more difficult to do. And so I was just wondering, potentially you could look for clustering. So of a gene that looks interesting, and then looking around that gene to see if there's anything else that is a possible. Yeah.
Yeah, I think looking for biosynthetic gene clusters is good. We're also looking at the transcriptomic data to see, do we have some genes that are being expressed that we just have no idea what they are? Um, that is kind of interesting. So why are they being expressed? What are they doing? Those may be targets for um, maybe plastic degradation. So if they're there on the plastic, they're being expressed at a higher rate, maybe we can test some of those as well, because again, that database doesn't have a lot of different proteins in it. So we need to kind of look outside of the hits that we have, because maybe there are some novel proteins that we've just never really seen before. So there is no way to identify it. So we would have to just test it, see what plastic we pulled it off of, maybe use that as our target plastic, and then clone those proteins or clone the genes to express those proteins and see if we get degradation.
Because yeah, what we've found is classically aromatic degradation genes are usually R clustered.
Hmm.
And the lignin degradation genes, sometimes they're clustered with other ones, but sometimes they're not.
Mhm.
Mm,
So yeah.
yeah.
Okay. Yeah. Really interesting.
So, I see now 1 minute before the 11 Central Time. I want to ask one technical or question. You
Mhm.
may or may not know the answer. So when you mention last approach, put I forgot the name of the device, but put it into the trench, and
The
then
C cube.
yeah, CQ, thank
Hmm.
you. And then attach some plastic over there.
Hmm.
And then how deep they're going, and then how to make that plastic attached to your device secure without detaching from the device, the CQ.
Yeah, so the C cubes that we have currently deployed are anywhere from 3,000 to over 6,000 meters down. So
Wow.
those
Okay.
were in the trench and also around the Mid-Cayman Spreading Center.
Uh-huh.
Those plastics were secured to the cube just using zip ties, because it was another form of plastic. So we were kind of
Uh-huh.
trying to
I
be
see.
able to sample anything that was on this cube and just see what we got.
Uh-huh.
They were placed by the Alvin submersible. It's not like we just dumped them off the side of the ship and let them sink. So
Mhm.
it was kind of a gentle, like setting it down in place. So those plastics weren't really jostled or anything. They were secure enough for the way that they were put there. But if you were to just kind of drop one,
Mhm.
you might need to secure them a little differently.
I see. I see. Because I
Mhm.
thinking about, entire plastic should be, securely attached to the device. Otherwise,
Yep.
we are also creating some more pollution to the ocean. And also, you're not gonna get the real good, sampling because that is
Good.
somehow, detached from the one. But that's a very interesting work. I know your work very well, so I'm not gonna ask more questions because I already asked many questions to you. But this is a fantastic work. Again, congrats again. Regarding your promotion. So I probably going to close now. Because after 11 Central Time. Okay, so thank you all for joining and staying today. We'll meet again on July 24, Thursday, the same time, the same Zoom link. We'll have Dr. Emma E., an editor at Cell, and also Professor Miguel Jimenez at the Boston University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank. I stop recording. Give me 1 second.